Guild. Now I would like to invite Professor José María Monserrat about nanomaterial risks for the aquatic environment. He's formed by the University of Buenos Aires, 1988. He made a master's in biostatistics at the same university, has has had doctorate in geography at Rio Grande. He's also a joint professor at the Biology Institute, has published several articles and 4 chapters in book. Biochemistry and animal physiology are his areas, and also aquatic toxicology. He has started new research about toxic components. He's permanent professor in post-graduation in physiological sciences, agriculture, and collaborator in chemical sciences and life and health. He's an assessor at scientific processes submitted to Brazilian and foreign agencies and has written articles in more than 15 journals. The Journal of Brazilian Toxicology and Chemistry, Ecology, and Open Toxicology Journal.
Good afternoon. Thank you for your invitation. I thank the Organizational Commission to help us present what our research group is presenting, and we're going to talk about risks and the nanomaterials for the aquatic environment. We're going to talk about some general aspects. We're going to talk about the photo excitement of nanomaterials and the future perspectives, which I believe that should be, as the professor said, we should see this quickly. All of us know that the nanoparticles are defined as entities that have a dimension between 1 and 100 nanometers. And from the ecotoxicological point of view, there are some variables as the ionic force, the salinity, as we're talking about the aquatic environment. There is a dynamics of an agglomeration that can certainly alter the toxicologic properties and also the compartment where these particles act. They can pass from the aquatic environment to the sedimentary part. So the technological bio properties of the particles are associated with their relative surface, and what is associated also is the potential toxic effects that it can present. Inside the nanoparticles, we have a division that is also operational. So we can talk about the engineered nanoparticles that are produced by man with specific and others that have anthropogenic origin, but that have a chemical variety which is bigger than the manufactured ones. Within the manufactured ones, we can differentiate the inorganic and the organic, and some of them are very interesting in the ecotoxicological, are very interesting. That would be the nano silver. We're going to talk about it. And in the organics, the carbon molecules like fullerenes and the nanotubes, as has been said, can be of simple wall or multiple walls. if we talk about the environment and we talk about the ecotoxicology, or as some say nanotoxicology, we should concentrate on how the investment is happening in this kind of research. The data that exist up to now show that, for example, in the United States budget for nanotechnology, the percentage that is dedicated for health and safety and also the environment, and it is a percentage. It is a layer that is not very significant of the whole budget. If we consider of what the European Union called framework programs, we'll see that in the different areas, this kind of project that is associated to nanotechnology, the ones that have to do with health and environment, represent a fraction that is not very significant within the thematics that are presented in this EFP program. And as in the same paper that they published in Brazil, they show a very similar tendency. There isn't a action that is directioned to assess health-wise the effects of the nanomaterials. It occurs more because of initiative than than being something that is organic, in my opinion. Even so, if we analyze that the ecotoxicological data exists, and up to now, a paper that was published this year shows some characteristics that are that this is as if it were a summary of the basic data and the ecotoxicological, in which we can see here different kinds of materials and nanoparticles that are organic and inorganic. And today what we see in yellow, it represents the group of organisms that presents the smallest value of 50, and the algae group seems to show the biggest sensitivity compared to others for a reduced number of nanomaterials, but I would still say that it is one of the few ecotoxicological work that we can make an essay of what we have now at this time, at this moment. Studies that have assessed the presence of nanomaterials that were engineered in the environment are very few. Very few exist of an analytic chemistry, but there are some papers that have started doing with studies of simulation in computers to assess which would be the ones that have a bigger risk or possibility of appearing in a bigger proportion or quantity in the environment. This was a work that was published last year in which there was a simulation with different kind of particles with oxide, titanium, and zinc oxide, and nanocarbon tubes, and fullerene, and you could see the concentration of these nanoparticles in the sediment. And this is in the order of milligrams per kilo, while the other other particles are in micrograms per kilo, so there is an idea of more accumulation. This is a simulation in the case of the titanium oxide when compared to other kind of materials. This maybe it will be one of the forms to approach, as what Dr. Weissman said. We have so many particles to work with. Which would be the criteria, the form to approach this problem? If you have a variability in molecular variability so big, if we consider nanoparticles that have been already incorporated in the human use, as typically the nano silver that is part of a tissue. Here we see that it is in materials, so it is present in stockings that can have more or less quantitative, or others that do not have nano silver but are sold as nano silver. What happens when they are washed? There is a release of silver and in the washing process, and the results appear that because there isn't a correlation between the concentration of silver in the stockings with the quantity of silver that is released in the washing. The method and technology of the fabrication seems to be determinant with how much is released when the material is washed. If we concentrate in the aspect of toxicity, a nanomaterial that has been very much studied is the fullerene. It has been much studied toxically. It is a molecule that has been synthesized. It can have 60 or 70 atoms of carbon, very little solubility in water, and it has it is easy to functionalize. It could incorporate carboxyl groups, and the solubility can increase drastically. But even so, the fullerene, sad as we see it here, it can form clusters in the water, and this will be smaller or bigger depending on the ionic force of the water. If it grows a lot, it can precipitate, and then we can consider another compartment that would be associated to the sedimenter. The biological organisms present biological associations that are interesting, and we can call generically called biofilms that represent an organic substrate that is friendly for the colonization of bacteria. And we know that there are many secretions and in mucuses of fish and nematoids and polychaetes, and they create this colony of bacteria, and as many materials represent effects that are bacteriostatic or killing the bacteria, with the material that we are developing, it is to assess what is the result of the effect of the bacteria communities that live on the surface of those organisms when they are presented with the fullerene. So we can see that the concentration of rose bacterium with different exposures to fullerene, we can see that some colonies that live on the surface of the fish, and they are reactive colonies, they increase the concentrate of ROS and of C60, while other colonies that are not. This is not verified on this in the same size, and the colonies that are being exposed to the 3 different concentrations of fullerene, indicating an effect, a bacteriostatic effect. So there wasn't a bactericide effect. And some of the colonies in which we isolated that we could characterize as C60 reactive or non-reactive due to this behavior of increasing or not the reactive colony, we could detect that there was a predominance of the general Aeromonas, and this is in a line of interest that is very to us, because now we're starting to work with zebrafish, with this colony of bacteria that are living on it, and how they utilize the nano silver. We have an Argentinian company that is manufacturing material with nano silver for material, and we are exposing this zebrafish to exposure of nano silver, and we try to study the bacterial growth. So what we observed is that in 3 different concentrations of nanosilver, there is a diminishing of the concentration of number of bacteria in the nanofish. But we also assessed not only the bacteria, but the answers of the organism in itself in terms of antioxidant capacity in the branchia or the brain, that we saw that when we have a bigger relative area, it has a lesser antioxidant capacity, but not on the branchias, but on the brain, there is a progressive loss of the antioxidant capacity, total one. And this is important because the communities, the biofilms, interact with the organisms in very complex ways with a big biological richness. So when we talk about the polycat, where this mucus offers this a superior concentration of hydroxy, so the mucus offers a competence of protection of antioxidant for the polycat. It benefits from this mucus that is friendly for determined bacteria to colonize. So what is important also is the functionalization of the fullerene. It can change its capacity and properties, toxicological properties, and it can pass from oxidant to a molecule that is antioxidant. We have made fluorometric assays that we used some molecules that can emit fluorescence, and what we want to see is that with these nanoparticles, how it can modulate a top going up or down this fluorescence. What does this mean? That the what generates radicals in the presence of nanoparticles can increase the fluorescence. So then the nanoparticles are being pro-accident. If it generates less fluorescence, this is because it is the nanoparticles is behaving in an antioxidant way. So we see that the pristine fullerene is pro-oxidant, and when it with hydroxic groups, it is antioxidant. What is interesting is that to have the antioxidant effect can be reversed in smaller concentrations. When you use mannitol, that is a scavenger of hydroxyl, what we can see is that there is a reversion in the fullerene that is total. We pass from the original situation of pro-oxidant to the situation of antioxidants. This tells us a lot of how we are thinking. We began now an approach in ex vivo using fish branchias in which the idea basic would be if it produces a quantity of oxygen, what can happen if it interacts with nanoparticles, other species of active oxygen? What will happen with this interaction? What would this have as an effect in the cells that are exposed to these nanoparticles? If we talk about the photo-oxidation of nanomaterials, the fullerene has happened for a long time. We know that in the presence of light with time, it increases significantly. Different kinds of forms to analyze and quantify conjugated DNs and T-bars and LOOH. So we'll see that part of this lipidic will be limited with scavengers, with the singlet oxygen, which means that light that is visible above the nanoparticles with fullerene can give way to singlet oxygen. This also can happen with zebrafish embryos, that we'll see that the larvae mortality is much bigger when you expose these embryos of the zebrafish in presence of light than when you keep them in darkness, which means that light is a factor that increases the toxicity. When you expose them to light, part of the mortality doesn't happen if you make a pre-treatment with NACT cysteine that serves to concentrate the concentrate. And light and fullerene are promoting a generation that are active to oxygen, increasing mortality. So for the future, a point that some colleagues have talked, does the same thing happen in the nanoparticular way? Does it happen also in the ionic form? Are we talking of the same particle? In case of the cell mortality, this work shows that it is not the same thing if it happens ionically or in the nanoparticle. If we talk about reproductive answers using the chlorate of 5, the answer is the same because there seems to be no difference between being or not a nanoparticle. And if we talk about the absorption dynamics of nanoparticles and of titanium oxide, we can see that the titanium oxide it absorbs arsenite and arsenite, and we'll see that when it accumulates in the branchias of the carps, we can see that it accumulates when there is an exposure to nanoparticles of titanium. So it's like a Trojan horse. There is some work that we're doing where we observe the presence of arsenito in the presence of fullerene. Nothing happens, but if we observe that the fullerene is avoiding this peak of reduced glutathione that are in the hepatocytes of the zebrafish, that are this has been already described, including for human cells. And if we analyze the peroxide lipidic in a small concentration of arsenite without fullerene, it seems to amenize the effect. It seems that fullerene plays against the toxicity of the arsenic, but in other combinations, for example, benzopyrene with C60, what you can observe here is the fluorescence, intracellular fluorescence that are seen in a cell of benzopyrene. We can see that with the fullerene, with time, there is an accumulation that is significant that doesn't happen when you expose the benzopyrene without the C60. If we observe a measure of functionality through the reduction of the MTT, we observe that the benzopyrene and different concentrations of it doesn't affect statistically the reduction of MTT in the hepatocytes in the zebrafish, but when it is exposed to C60, there is a significant reduction that suggests a loss of the activity of the dehydrogenases. And if we analyze the transferase enzyme, we'll see that there is an increase in the hepatocytes in the benzopyrene, and this answer is abolished when the exposure happens simultaneous to the fullerene. So there is a multiplicity of factors that can be interacting in the nanoparticles that can diminish the toxicity of this molecule. Interactions of nanoparticles with other toxic moleculars that can even increase for them to enter the process. So and how to make them leave the aquatic environment and make them go to the sedimentary environment, and they can have a pro-oxidant response and also have a smaller relative area. So the polychaetes that were exposed to the sediment that was enriched with fullerene, they had more antioxidant characteristics. I would like to thank this possibility and very much the nano students and researchers who work with us together, and to all the agencies and institutions that who help us financially and logistically, and thank you very much.